Hello everyone and welcome back to our channel MooTechie. It's fantastic to have you here as we dive deeper into the incredible world of biochemistry. Today, we're connecting the dots between different cellular compartments to see how our cells manage energy and waste. It's a fascinating journey from the cytosol to the mitochondria. Now, let's set the stage with a big picture view, our metabolism map from the cytosol to the mitochondria. We have two main compartments illustrated here. On the left, the cytosol, and on the right, the mitochondrial matrix. In the cytosol, we see several key processes listed. Fatty acid activation catalyzed by acyl-CoA synthetase and part of the urea cycle involving the enzymes ASS, ASL, and arginase. Inside the mitochondrial matrix, we have beta-oxidation and the enzymes CPS1 and OTC. Connecting them is the carnitine shuttle involving CPTI1 and CPTI2, which transports fatty acids into the mitochondria. There are also specific carriers for moving ornithine and citrulline across the inner membrane. The summary text explains, in the cytosol, fatty acid activation and some urea cycle enzymes operate. In the matrix, beta-oxidation and other urea cycle steps occur. Transport is facilitated by the carnitine shuttle for fatty acids and special carriers to exchange ornithine and citrulline. Let's break down the first transport system. This slide details fatty acid activation and transport via the carnitine shuttle. Step 1 is activation in the cytosol. The enzyme acyl-CoA synthetase converts a fatty acid plus coenzyme A and ATP into acyl-CoA synthetase. Step 2 is outer membrane entry. The enzyme CPTI1 or carnitine palmitoyl transferase 1 converts fatty acyl-CoA and carnitine into acyl-carnitine. A crucial regulatory point here is that malonyl-CoA inhibits CPTI1. Step 3 is transport across the inner membrane. The carnitine-acyl-carnitine translocase shuttle moves the acyl-carnitine into the mitochondrial matrix. Step 4 is matrix release. Inside, CPTI2 converts the acyl-carnitine ACH into fatty acyl-CoA and free carnitine. The slide also has a helpful compartment flow diagram. It shows the cytosol and the matrix with the pathway CPTI1 to the translocase to CPTI2. The inhibition of CPTI1 by malonyl-CoA is prominently noted. A color key is provided. Blue represents enzymes, green represents carnitine, and purple represents acyl-carnitine and other intermediates. Moving forward, we reach the core breakdown process, the beta-oxidation spiral, which consists of 4 recurring steps. Step 1 is oxidation or dehydrogenation. Acyl-CoA dehydrogenase creates trans-delta-2-enoyl-CoA and reduces FAD to FADH2. Step 2 is hydration. Enoyl-CoA hydratase adds water to form L3-hydroxyacyl-CoA. Step 3 is a second oxidation. L3-hydroxyacyl-CoA dehydrogenase acts, reducing NAD+ to NADH. Step 4 is thiolysis. Beta-ketothiolase with another CoA cleaves the molecule to produce one acetyl-CoA and a new shorter acyl-CoA that is 2 carbons smaller. The outcome per round is clear. 1 FADH2, 1 NADH and 1 acetyl-CoA is produced and the fatty acid chain is shortened by 2 carbons. A special note is made for odd-chain fatty acids. Their final round produces propionyl-CoA, which is then converted to succinyl-CoA, a TCA cycle intermediate, via a pathway requiring biotin and vitamin B12. The color key here shows blue for enzymes, purple for the energy carriers FADH2 and NADH, and orange for acetyl-CoA. Now, let's talk numbers. What's the actual energy yield from beta-oxidation? Our example is palmitate, a 16-carbon fatty acid. The net yield is 106 ATP, calculated as 28 ATP from the electron transport system plus 80 ATP from the TCA cycle minus the 2 ATP cost for initial activation. This uses modern PO ratios where each NADH yields about 2.5 ATP and each FADH2 yields about 1.5 ATP. Let's walk through the calculation. For palmitate, 7 cycles of beta-oxidation yield 7 FADH2, 7 NADH and 8 acetyl-CoA. Oxidative phosphorylation via the ETS yields 7 times the sum of 1.5 and 2.5, which equals 28 ATP. The 8-acetyl-CoA molecules enter the TCA cycle, each yielding about 10 ATP for the total of 80 ATP. From this, we subtract the 2 ATP cost of the initial activation step. So, the summary is 7 cycles produce 7 FADH2, 7 NADH and 8 acetyl-CoA. The TCA yields 80 ATP. The ETS yields 28 ATP. After the 2 ATP activation cost, the net ATP is 106. Next, we shift from energy production to waste management with an overview of the urea cycle and its compartments. The diagram clearly splits the cycle between the mitochondria and the cytosol. In the mitochondria, we find the enzymes CPS1 and OTC. In the cytosol, we find ASS, ASL and arginase. Ammonia, NH3, enters in the mitochondria, and urea is the final product released from the cytosol. A molecule called fumarate is also produced, which can feed back into the TCA cycle. The text explains the cycle's purpose to detoxify ammonia into urea for safe excretion. It spans 2 compartments. The mitochondria house CPS1 and OTC, while the cytosol houses ASS, ASL and arginase. Special exporters and importers move citrulline out to the cytosol and ornithine back into the mitochondria. The link to the TCA cycle is important, as the byproduct fumarate connects the urea cycle to central metabolism. Let's look more closely at the urea cycle enzymes and nitrogen sources. The diagram again shows the mitochondrial and the cytosolic compartments. Key mitochondrial enzymes are CPS1, activated by NAG, and OTC. Key cytosolic enzymes are ASS, ASL, leading to arginase. The text notes the enzymes CPS1, OTC, ASS, ASL, and arginase. The nitrogen sources that feed the cycle are free ammonia and the amino acid aspartate. N-acetyl-glutamate or NAG is a critical allosteric activator of the first enzyme CPS1. Transport across the inner mitochondrial membrane is facilitated by specific carriers for ornithine and citrulline. Understanding how this cycle is controlled is crucial, so let's discuss urea cycle regulation and clinical notes. Under regulation, N-acetyl-glutamate or NAG activates CPS1. The amino acid arginine increases the synthesis of NAG. The rate of the cycle is driven by substrate availability. More ammonia and aspartate increase the metabolic flux. Under clinical notes, a deficiency in OTC or ornithine transcarbamylase is an X-linked disorder that leads to hyperammonemia and an increase in orotic acid. Therapy options include sodium benzoate, sodium phenylbutyrate, lactulose, and moderating protein intake. In a crisis, dialysis may be necessary. Now, let's connect amino acid metabolism through transamination and the PLP mechanism. The key reactions are ALT or aligned transaminase converts alanine and alpha-ketoglutarate to pyruvate and glutamate. AST or aspartate transaminase converts aspartate and alpha-ketoglutarate to oxaloacetate and glutamate. Their cofactor is PLP, pyridoxal phosphate, a derivative of vitamin B6. They operate via a reversible ping-pong or double displacement mechanism. The slide elaborates. ALT moves nitrogen from alanine to glutamate, linking muscle metabolism to the glucose-alanine cycle. AST supplies aspartates nitrogen to the urea cycle via oxaloacetate. The PLP ping-pong mechanism involves the amino group transferring to and from the PLP and PMP intermediates, allowing the enzyme to be reused. The summary points are ALT and AST are reversible and require PLP. Glutamate acts as a nitrogen funnel, directing nitrogen towards deamination and the urea cycle. The transfer uses ping-pong chemistry. Clinically, elevated AST and ALT levels suggest hepatocellular injury like in liver disease. The body needs safe ways to move ammonia around. Let's explore ammonia transport and detoxification. The diagram shows tissues and cytosol on one side and the liver mitochondrial matrix on the other. In tissues, glutamine synthetase traps toxic ammonia by converting it into glutamine, a safe transport form. The ALT reaction in muscle forms alanine as part of the glucose-alanine cycle. The brain also detoxifies ammonia primarily via glutamine. Glutamine travels to the liver. In the liver mitochondrial matrix, the enzyme glutaminase releases the ammonia, which then feeds into CPS1 of the urea cycle to make urea. The glucose-alanine cycle shuttles nitrogen from muscle to the liver as alanine. In the brain, detoxification via glutamine is vital because excess ammonia can lead to cerebral edema.
This is where it all comes together. Let's look at the integration of lipids, amino acids, and the TCA cycle. The diagram is rich with connections. In the cytosol, we have transamination producing aspartate and the aspartate arginosuccinate shunt. Beta-oxidation in the matrix produces acetyl-CoA and NADH/FADH2 for ATP. The urea cycle uses aspartate and releases fumarate. Text explains transamination supplies aspartate for the urea cycle and the urea cycle byproduct fumarate feeds into the TCA. During fasting, malonyl-CoA levels drop, which relieves inhibition on CPT1, allowing more beta-oxidation. Low levels of oxaloacetate can also shift metabolism towards ketogenesis. A legend decodes the diagram. Beta-oxidation leads to acetyl-CoA and reducing equivalents that make ATP via the TCA. The flow between mitochondria and cytosol involves the carnitine shuttle and the ornithine/citrulline carriers. As we wrap up, here are the key takeaways. First, for beta-oxidation, remember the carnitine shuttle and the 4-step spiral. For palmitate, the net ATP yield is 106. Second, transamination uses PLP, ALT and AST funnel nitrogen to glutamate. Third, in the urea cycle, CPSI is activated by N-acetylglutamate or NAG, and the pathway cleverly spans both the mitochondria and cytosol. Fourth, ammonia detoxification employs multiple strategies. Glutamine synthetase makes glutamine, the glucose-alanine cycle shuttles nitrogen, and the urea cycle completes the process. A color guide is provided. Blue is for enzymes and beta-oxidation, purple for cofactors and mitochondrial elements, green for cytosolic metabolites, red for ammonia, and orange for transport and energy links. The slide is signed by MooTechie. Finally, we have our closing slide. MooTechie, thank you. Stay connected with MooTechie. Keep learning, keep exploring biochemistry with us. Thank you. See you in the next class.